All right, hello and welcome to the pre-lab slide for week 5, which is the last week of this very long LDH lab. So just to remind you what we've done and what we're going to do this week, for the first 2 weeks we started purifying our LDH from our beef extract, or our beef heart. And once again, I forgot to correct this slide. So of course we used beef heart, not potato. So to purify our LDH, we use several techniques, including homogenization, salting out, centrifugation, and dialysis. In week 2, we finished purifying our LDH and started preparing some of our samples for analysis. And we did this by using 2 main techniques. We used affinity chromatography to help purify the LDH from our dialysate. And we also used a desalting spin column to help remove some of that ammonium sulfate from our salting out sample. In weeks 3 and 4, we started to characterize our purified LDH. And we did that using our 2 assays. First, we did our Bradford assay to quantify our total protein. And then we did our activity assay to measure how much active LDH we had in each sample. So that would help us calculate the specific activity of LDH in each of our samples. And finally, this week, we're going to finish up characterizing LDH by doing some gel electrophoresis. So let's talk a little bit about the theory behind gel electrophoresis. So in general, gel electrophoresis is a technique we can use to separate molecules by their size, shape, and charge. Here we use several reagents to help us make our gel electrophoresis assay only separate our proteins by their size. So let's talk a little bit about how we do that. So first, the gel is made of several components. The main component is polyacrylamide. Polyacrylamide essentially makes a gel sort of matrix that helps to trap proteins inside. And so larger proteins are impeded by traveling through the gel, by the gel itself, and smaller proteins are able to travel through more of the pores in the gel, and they're able to travel towards the bottom. So overall, we would see smaller proteins travel to the bottom. We would have larger proteins that are trapped near the top. And so that's how we're going to be separating our proteins by size. So the real question is, how do we eliminate the effects of the shape and the charge of our proteins? So we do that by the next couple of ingredients. So one of the important ingredients we use is a detergent, and the one we use is called sodium dodecyl sulfate, or SDS. So that has a couple of purposes. So first of all, SDS is itself charged. It has a negative charge, and it's going to coat and surround each of your individual protein molecules. And so by doing that, you'll get this uniform negative charge density around your proteins. So it doesn't matter what the original charge of your protein was. Once you coat it with SDS, it's just going to be negative. And so that's what makes proteins attracted to the anode, which is located at the bottom of the box where we run our gel. The other thing SDS helps to do as a detergent is to help unfold our protein. And so this helps to eliminate some of the effects of the protein's original shape. So overall, SDS helps to make our proteins linear and uniform and have a uniform charge density. So this is the main thing that helps us only separate our proteins now by their size. The other important ingredient we use for this is a reducing agent. And the one we're using today is called beta-mercaptoethanol, which we often abbreviate BME. So BME's main job is to reduce bonds, and specifically we're looking for it to reduce disulfide bonds that might be present in some proteins. And again, this just helps to make sure that our proteins are linear, they're unfolded, so that we don't have this effect of the protein shape as it's traveling through the gel. And then the last thing we need to do once we're done running the gel is to stain it so we can actually visualize our proteins. And the stain that we use is called Kumasi blue. And Kumasi blue sticks to proteins. It doesn't stick to the gel very well, and so you'll find that after you stain, all the proteins in your gel will be this nice kind of bluish purple color, and the background of your gel will be pale blue or clear. And so you'll be able to see all of the proteins that are in each of the wells of your gel. And as a reminder, our big molecules, that is bigger proteins, are going to move smaller distances. So they'll be trapped near the top of the gel where we loaded our sample by the cathode. And the smaller proteins will be able to travel further and faster towards the anode as we run the gel. So let's get into what results we might expect from our purification process in this sort of theoretical gel we're going to look at here. So this theoretical gel we're looking at now has more samples in it than you will be running. You will only be running, I think, about 9 samples. Here we have more than that. But anyway, let's look at each of the different steps we would have seen along the way in our purification process for LDH. So over here on the left, we're showing a ladder. So a ladder is going to contain several proteins of known specific sizes, and they'll come out as nice, clear bands. They may be evenly spaced. Some may be closer to each other, some may be further. But this ladder is what you'll use as basically a sort of size standard to help you see the sizes of the proteins that you don't know their identities in each of your samples. So you can, given that we know the size that LDH is supposed to be, now you can look for the band in your ladder that's a similar size to LDH, and then use that to line up with your unknown samples to get a pretty good guess of which protein might be LDH and which protein might not be. And so just to be clear, this only tells you by size what a protein is, so it can't confirm a protein's identity. There might be other proteins in a sample of a similar size to LDH, and those would look the same on this kind of gel, because all we can do, once again, with this gel is to separate proteins by their size. So anyway, that's our ladder. And so just for the purposes of this exercise, we'll pretend that this band on our ladder corresponds with the same size as LDH. This may be different in real life when you run your own gels with your own samples. So let's think about our next sample in here, which is the homogeneous supernatant. So the homogeneous has a big mixture of proteins in it. It's completely unpurified, so what you will likely see here is just all of the different proteins from the contents of the beef heart, and so you'll probably see what looks like basically a smear. And you might see some darker bands in your smear. You'll probably see a band for LDH. You may or may not, but you'll see a variety of proteins in your homogeneous. You probably won't be able to make out distinct individual bands, although you might see some darker and lighter areas for proteins that are more prevalent and less prevalent within the sample. So the next purification step that you did was your salting out step. So in our first salting out, we pelleted out proteins that were less soluble than our LDH. And so in your salting out one pellet, you might have, again, a variety of proteins. Here you'll probably see distinct bands. You shouldn't see a band for LDH in your salting out one pellet because your LDH should have stayed in the supernatant during this purification step. So as a reminder, these are the less soluble proteins. In the supernatant from your salting out, this is where we should have seen some LDH, right? So you should see a band for LDH in our imaginary gel here. And you'll also see a number of other proteins, all the proteins that are as soluble as or more soluble than LDH. For our second salting out, this time we pelleted out all of the proteins that were of similar solubility to LDH, and what remained in the supernatant were all the proteins that were more soluble than LDH. So what might that look like? So in our pellet, we would of course see a band for LDH. We would see some of these proteins maybe have similar solubility. And we would see some of these proteins probably are more soluble, so they'd remain in the supernatant. See if I did that right. Yeah, so you'd see So I drew these lines here this way, these bands, because we're starting out with the supernatant from our first salting out. So we'll see all of these bands and they'll be split. Some of them will have stayed in the supernatant and some of them will have gone into the pellet in our second salting out. So at this point, from our salting out to pellet, you did a dialysis to remove some of the extra salts from your buffer. But dialysis, the pores in the dialysis membrane are too small to allow proteins to go through. So your dialysate, the protein content should be the same as this salting out to pellet. Because the protein content of these hasn't changed, although the buffer and salt composition may have changed. So that brings us to our affinity column. So we did a few different steps in our affinity column and collected samples from each of those. And so your affinity column, when you ran it, the LDH should have stayed stuck to the column and not come out until you started eluting. All of your other proteins that we're seeing in our dialysate sample should come out in the flow-through. So our flow-through should look a lot like our dialysate, except missing our band for LDH. Our wash should look very similar to our flow-through, except maybe a little less concentrated. So you might see nothing in your wash, or you might see very faint bands at these same sizes from your flow-through. And then finally in your element fractions, which are here labeled fractions 1 through 8, that's where you're going to see your LDH. So maybe you got your LDH in fraction 2 and fraction 3 and fraction 4, just for an example. So you might see very strong LDH bands in those particular samples, and you might see no proteins in your other element sample fractions. So what does this mean for you when you're doing this lab? So these are the samples that you're going to be running on your gel that you should have, saved in vials in the freezer. So you'll be running your homogeneous sample, your retentate from your desalting column, your dialysate supernatant, your flow-through, and your wash. And you're going to then pick out 4 element fractions. I'd like you to pick out the ones that have the highest concentration of LDH or the highest concentration of protein that you saw in your Bradford, either one. Just to see, hopefully that way you'll actually see bands on your gel and not just have empty lanes. And of course you're going to want to put a protein ladder in your gel. And so I should mention on the previous slide when we had our protein ladder, it was all the way on the left-hand side of the gel, just for convenience in explaining. In real life, when you run your gel, it would make more sense for you to put your protein ladder somewhere near the middle of your gel. And this makes it easier to compare the band sizes that you see in your ladder with the band sizes that you see in all of the other samples that you're running in the other wells of your gel. So how do we prepare our samples? So just to start off, the Kumasi stain is a little bit less maybe sensitive than your Bradford assay. And so you don't need to dilute your samples unless that sample in your Bradford assay needed to be diluted more than 100 times, or at least 100 times. So if you only required a 1 to 10 dilution for your Bradford, you do not need to dilute your sample to run it on the gel. So once you have your sample or your dilution prepared, you'll mix 15 microliters of your sample and 15 microliters of sample buffer in a 1.5 ml tube. You do not need to do this for the ladder because the ladder already comes with the sample buffer sort of mixed in with it. So let's talk about the components of our sample buffer. So the first component is tris HCl. This is the actual buffer. This helps to maintain the pH of our sample. The second component of our sample buffer is STS, which we already talked about the purpose of. That's our detergent. It helps to keep our proteins linear and with that uniform charge density. The next component of our sample buffer is 10% glycerol. So glycerol is denser than water. It's relatively dense, and so it's denser than our buffer. And so what the glycerol helps with is making sure that your sample will sink to the bottom of the well, and your proteins don't start floating away somewhere before they get pulled into the gel. The next component is more BME. We already talked about how BME is a reducing agent that helps to break disulfide bonds and keep your proteins linear. And then the next component is EDTA. Oh, and before I talk about EDTA, I just want to mention for BME, it has a very distinct odor. When you are using sample buffer, when you open the container, the odor will spread throughout the room potentially. So whenever you're opening a container of sample buffer, please keep it open for as little time as possible. Put the cap back on as soon as you're done. Any containers that you put anything containing sample buffer into, again, could be smelly. So please try to keep any containers that you put sample buffer into or that contain sample buffer, if you open them, try to close them as soon as you're done, as quickly as possible, just to keep in mind everyone's sense of smell, just to save everyone from experiencing that unpleasant odor. We will have the hoods running in the lab, and that will help with some of the smell, but it's very powerful, which you'll see. Okay, so that brings me to EDTA. So EDTA is a chelating agent. It helps to bind divalent cations in solution. It also just, in general, its function is to sort of help keep your protein nice and reduced. It'll bind any extra ions that are floating around in there that might potentially add charge to your protein and maybe alter the charge or the shape, which we don't want to do, right? Because we're trying to have uniform shape and uniform charge. We want the only differences between our proteins to be their size. And then finally, the last component of our sample buffer is this bromophenol blue dye. So bromophenol blue is a great dye because the way it will travel through the gel is it will travel before all of your proteins. So you'll have what we call a dye front. So it'll look like this blue line that's starting to travel down the gel. And when that blue line reaches the bottom of the gel, that's when you know your gel is finished running. And because it runs in front of even the smallest proteins, that will help you make sure that you don't accidentally let any of your protein samples go all the way past the bottom of the gel and out into the box where you wouldn't be able to see them anymore. You can't see the proteins, of course, while they're running. You can't see them until we stain our gel, which is after the gel is finished running. So that's why we have this bromophenol blue dye in the sample buffer itself. So let's talk a little bit more about the gels that we're going to use. So in this slide, we have a picture of the two sides of a gel. They are prepackaged, they're prepared for you. And so what these consist of are, there's two plastic plates that are very thin, and sandwiched in between, in the middle of them is your actual gel. It's very, very thin. And on the top of these two pieces of plastic with the gel inside, you have this gel comb that's green. And so it's really, you see well numbers labeled here, right? You see number 1, 2, 9, and 10 labeled on the left. On the right, you can see more easily all 10 of those wells. So that's what the teeth are on those combs. So those teeth sticking into the gel means that when you pull this comb out, you'll have nice little well shapes in your gel. And when you insert your sample, after you take this comb out, you'll insert your samples into each of these wells that are in the gel. Something important to pay attention to with the pieces of plastic that contain your gel. It's easier to see in this picture on the left, but there's what we call a long plate and a short plate. So the tall plate behind here is what we call our long plate. It extends all the full height of the gel. There's also a short plate. The short plate is sort of in front of the gel. And so you have this, you have one long plate, one short plate. It's really important to pay attention to this when you put your gel into the gel stand. You want the short plate to be touching the gasket on the gel stand. So it's essentially facing the inside of that inner reservoir. And this is going to be important for when you actually load your samples and for letting your gel run. Your gel won't run if you have it the other way because you need the buffer in that internal reservoir to be touching the gel. And that's only possible if the short plate is facing the inside. The other very, very, very important thing I want to talk about here is the sticker. So it's not shown here, but you'll have on your gel, when you pull it out of the package, there is a green sticker that is going to be on the bottom of the plastic plate. Please, please, please take the sticker off before you put the gel in the gel stand. Your gel will not run if you do not take the sticker off, or if it does run, it will run unevenly and not give you good results. So I know it's not pictured here, but please, please take off that sticker before you do anything. So let's talk about the next step, which is the gel stand. So the gel stand is where you're going to be putting your gels into. So you can see here we have this green gasket. Let me just change colors real quick. So we have this green gasket, and this outline basically outlines the shape of the plate where your gel is. And so once again, when you put your gel in here, you want the short plate to be touching that green gasket. So the long plate is facing you, the short plate is facing sort of the inside here. And you want to kind of push the gel so that the bottom of the plastic lines up with this piece of plastic on the bottom of the gel stand here. And then once you have your gel loaded in, you'll take these green arms and rotate them back up, and that helps to lock the gel in place. We also have a couple of gel stands that look a little bit different from this picture. And if you end up using one of those, we'll show you how to do that when you get to the lab. So depending on, okay, so the way these gel stands work is you need to always have two different gels on them, one on either side, and this creates this enclosed space in the middle called the inner reservoir. And so you'll load your samples into one gel. You can, your group, your partner, or yourself, depending if you're an individual or partner, you can team up with another group, and you can each put your gel on either side of this gel stand. You need to have something on each side of the gel stand, whether it's someone else's gel or an old expired gel that we just won't load samples into. But the gels, again, they won't run if you only have one gel in your gel stand. You need two. So either find another group to pair with, or ask for an old expired gel that we can put on the other side of your gel stand. So once you have your gels all loaded into your gel stand, then we can put that whole stand into the gel box. So, and everything in here is color-coded to hopefully make it very straightforward for you. So on the gel box itself, we have these black and red bars. These are going to line up with the electrodes on your gel stand, which are also labeled in colors in those black and red. And again, you want them to match. In this picture, we only show one gel on the gel stand, but in real life, when you put this in, there will be a second gel on the other side of this gel stand. And then what you want to do once you have your gel in your gel box is you can start to fill the internal, and once again, there will be, a second piece of plastic here. And once you have everything loaded in, what you want to do is put the running buffer into this space that you've created here inside the gel stand. So you can fill this with running buffer, and you want it to be filled pretty high, almost to the top. So you want it to go past, so like your short plate is facing the inside here. You want the buffer to go past the top of that short plate, and it can go up to the top of the long plate even. Once you filled up this internal reservoir, you want to leave the gels just sitting there for a couple of minutes and check for any leaks. If you have leaking at this point, you're going to have a problem running your gel later, because as you leak, the gel will leak out from the internal part here to the external reservoir here. And once the buffer level drops too low, once it goes past the top of your short plate and it's not touching the gel anymore, your gel will stop running. Which is a problem. So before you get anything started or load your samples, make sure you do not have any leaking from this internal reservoir. And once it's filled up with buffer, you can also remove the gel comb because you can't load your samples until you take the comb out to expose the wells. All right, so like I said, now at this point, you've made sure there's no leaking, you're ready to load your samples. So you'll remove this gel, this green comb, and you want to do this assertively but carefully. You want to slide it straight up. You don't want to wiggle it from side to side, because that can move around these little kind of arms that separate the wells. You want those kind of well separation pieces to be vertical, so you can load your samples in between each one of them. So it may take a little bit of force to pull that comb out, but be careful and just pull it straight without wiggling it. And then you will take a gel loading tip. and put it onto a micropipette so that you can load 20 microliters of each of your samples as well as your ladder into the gel. What's special about these gel loading tips is they have a very narrow end. This makes it easier to actually stick it into the wells. So remember, like we said before, the gel is located in between these two pieces of plastic, in between the short plate and the long plate. So when you have your pipette with your sample, you want to stick that pipette tip in between the two plates and directly into that actual well of your gel. You don't want to, and you want to make sure you don't miss it, right? You want to make sure you get the pipette into that space and not in front of it or behind it. And that's one of our cautions, so don't miss the well. The next caution I will have for you is do not puncture the well. So when you put that gel loading tip in, you want the end of the tip to extend maybe halfway down the well. You want it to be in the well, but you don't want it to be touching the bottom of the well. If it touches the bottom of the well, you have a risk of accidentally sticking your sample too far into the gel itself and not into the well. And that can cause problems. You can, you don't want to do that. You want your well to be, you want your sample to be sitting in this well. You don't want to put it anywhere else, otherwise you could get unclear or improper results. And finally, be careful not to release air bubbles. So if you have air bubbles that you push out along with your sample, the air bubble could displace your sample by, if you shove bubbles in here, your sample could end up floating out into the space above the well, which again could be a problem. So so those are the 3 cautions. Please make sure your tip ends up in the well of the gel and not in front of it. Do not puncture the well by pushing the tip in too deeply, and only push the plunger of the pipette far enough to dispense your sample without releasing air bubbles. So once your samples are loaded into your gel, now you're ready to actually start running the gel. So what you'll need to do at this point is put the lid onto the gel box, and again the lid has the 2 wires for positive and negative, and the colors are going to match the colors that are on the box and on the gel stand. And then you'll plug in the other end of the lid into your power supply, which again is color-coded. So just to keep in mind with your power supply, it's perfectly okay to stop and start as needed. So if one group or set of groups is ready to run their gel, they can get started. If someone else needs to join in with that power supply, you can just pause the run, plug in the new one, and restart the run. Just be careful when you are running your gel to keep track of how much time your gel has been running, since all the different boxes that are plugged into the power supply will probably end up being run for different amounts of time. And the same thing if someone's gel is finished, you can pause the run on the power supply, unplug their gel box, and then restart the run with the power supply. We should be able to have 2 power supplies in the lab. So everyone will be sharing power supplies, along with probably also sharing gel boxes. Okay, so let's talk about running the gel. What do we expect to see when the gel is running? So when we first start, something that tells us that the gel is going is we'll start to see bubbles appearing, starting from the top and floating up through the inner reservoir. And this is a sign that the electricity is working on some of the water molecules in there and we're creating air bubbles. So that's a really good sign that everything is going well, is that you'll see bubbles. The other thing that you should start to see, especially after a few minutes, is the movement of the dye front. So you can see in this picture you have the dye that's sort of just below the level of where the wells were, and you see this line of blue colors. And so you'll see that line of blue gradually start moving down the gel. And once you'll know your gel is done, once that dye front reaches the bottom of the gel, which is down here. So if you're not seeing these 2 things happening, if you're not seeing bubbles and if you're not seeing movement of your dye front, here are some things you can look for. First, make sure, did you remove the tape, that sticker, from the bottom of the gel before you loaded it into your gel stand? If you didn't, your gel will probably not run. Did you check for leaks? When you put your gel into the gel stand, did you first fill that internal reservoir and see if the buffer stayed inside. Like I mentioned, if the buffer gets too low in that internal reservoir and the gel is no longer making contact with the buffer, you can't get the conduction of electricity, and the gel will not run. The proteins won't move. Next, sort of related to that is the buffer level above the top of the inner gel plate. So even if you don't have leaks, if your buffer level is not high enough on that internal reservoir, again, the gel won't be making contact with buffer and the gel will not run. And finally, the last thing you should make sure of is, are the electrodes connected correctly? So make sure red is red and black is black, and that way your charges are pulling the proteins in the right direction. All right, so once your gel is done, once your dye front has reached the bottom of the gel, now you're ready to remove it. So you can take the gel stand out of the gel box, rotate out those arms to free the gel, and now you have to actually open up those plastic parts that are surrounding your gel to free the gel itself, which again is sandwiched in between these two pieces of plastic that we're looking at here. So the way we do that is with one of these spatulas. So what's holding together the two pieces of plastic is actually just another little tiny thin piece of plastic. And so what you can do is essentially put the metal part of the spatula into the edge in between the two pieces of plastic, and use the spatula essentially like a lever to pry apart those two plates. And you might have to do this several times, going around the edge, especially at the corners, but eventually the two pieces of plastic will be able to separate and you'll be able to get your gel out. I also just want to caution you to be really careful here not to tear the gel. The gels can tear fairly easily if you're not careful with them. So make sure you're always wearing gloves. Make sure it sometimes can help if your fingers are a little bit damp when you're handling the gel. And when you're pulling apart the two pieces of plastic that are that sandwich around the gel, be very gentle. Do it very slowly. Allow the gel will probably adhere to one of the pieces of plastic, either the short or the long piece. So just let the gel go where it wants to go, and then gently take that gel and put it into a petri dish, which we see over here. And we'll put it in the petri dish for the next part of this experiment, which is staining the gel. So the staining is going to allow us to visualize the proteins that we've run on our gel, and we're going to do that in 3 steps. First, we're going to start with a rinsing step to remove any excess buffer or salts. The next thing we're going to do is do the actual staining, and this will turn the whole gel is going to look blue at this point. So the background parts of the gel will take on some dye, and the dye in particular will stick more strongly to the actual protein bands that have run down the gel. And so at the end of staining, everything will look blue. So that's why we have the last step, which is destaining. And during destaining, the dye will come out more easily from the gel in the background that doesn't have protein in it, and the dye will stay stuck onto the actual bands of protein. And so what you'll end up with will look a little bit like this. And in this picture, we can see our marker on the left, and then these protein samples in the other 5 lanes of this gel. In terms of recording the results, you'll want to take a picture of your gel once it's destained, so you can visualize all those bands and reference them later when you're writing your lab report. So you can do that a couple of different ways. You can use a light box to help see the bands more clearly on your gel. It's not always easier to see or take a picture with this light box. A lot of times people find it easier just to actually put the gel onto a white background, for example, a paper towel, and then just take a picture that way. Either way, make sure you take a picture of your gel before you leave. So at that point, once you have your gel destained and you have your picture, then you can interpret the information that you get from your gel. So the first thing you'll want to do is assign molecular weights to the protein ladder that you ran through the gel. And at that point, once you know the molecular weight, you can figure out which bands correspond with LDH based on their size in each of the samples that you ran in each of the different wells of your gel. And so at that point, you can start to ask, are your results consistent with the expectations, like the theory of this experiment, what you would think is present in each of your different samples along the way during our purification? And you can also ask if the amount of protein you're seeing and which proteins you're seeing are consistent with your Bradford assay and your activity assay, which should help you know how much LDH and how much total protein is in each of your samples. And if there are any inconsistencies with your expectations in any way, you can start to ask which results that you got are more reliable or less reliable. Like which results can you trust and which results maybe are less trustworthy. So with that, in summary, after this week, you will have visualized all the proteins that are present at all the different points in your purification process. You can estimate the molar masses of those proteins that you saw because of the ladder. And finally, you can qualitatively assess the relative amounts of those proteins just by looking at the density of the band. So a thicker or darker band has more protein than a very faint, weak band. And finally, at this point, you are done with this lab, not with the class, but with the lab. So at the end of week 5, you can finally discard all of your samples. So with that, good luck.